Welcome to 52 Living Ideas. Today, we will go back to a topic that we had begun last time, and it's about emergence. And we had talked about this in the past, but one session is we found, or what I found is it's definitely not enough. This is a very complicated topic. So I felt important to go into this a second time, and we may need to do it again, depending on the interaction and the level of understanding. But this is one of those areas of neuroscience that I feel is very, very important for people. It's one in the description of the talk tonight. I gave several areas that are important, and this is one of those. And like I said, we began this last month talking about it. But last month we looked at it from the point of view of doing a comparison, a compare and contrast with artificial intelligence systems, non-biological systems that have been appearing in the media and in the contemporary world. And we're trying to compare human behavior, animal behavior, and various levels of animal consciousness with what appear to be AI behavior and intelligence. And they're not conscious yet. Some people think that they will be. I'm one of those that believe they eventually will show many aspects of consciousness. I don't want to say if it will be consciousness, but it will appear to us as consciousness. So that was last month. Today we're going to look at it from a completely different area. And there are two things that I wanted to present, although I don't have a lot of slides on these, but we'll talk through them. One is we had talked about something about a year ago, which it's an organism that we had covered, which I have difficulty pronouncing it, placozoans. They have existed for a long time. They're multicellular organisms, and the way that they behave, the way that they function, it's very unique to the topic that we're talking about and how a lot of sophisticated behaviors emerged. They don't have a brain, but they seem to behave in somewhat intelligent ways as some more sophisticated multicellular organisms do. Even though they have no neural tissue, they have no neurons at all. So that's one area. And another area is something that we haven't covered, but it's actually research that has been ongoing for a few years by a professor at Georgia Tech. And there are new findings that he's written about, his team has developed. And it's on the concept of multicellularity, basically how unicellular organisms emerged out of, or multicellular organisms emerged out of unicellular organisms. For example, amoeba is a unicellular organism. It's basically a single cell. It lives by itself. It doesn't attach to other cells or organisms. It's basically a single cell. And that's the entire organism. But all complex life that we know of are multicellular. They have more than one cell. Human beings have, orders of multiple trillions of cells. And most animals have, again, small number of trillions or hundreds of billions of cells. And the simplest organisms, multicellular organisms, have tens of thousands of cells. So multicellularity was a very important issue that developed early in evolution. And that's something that we will talk about in this. So what I want to do, I'm not sure if everybody was participating in last month. So we have new people coming. So I'm going to just briefly touch on some of the things that we talked about, just in terms of what emergence is. This is important to talk about. And let me just try to, I need to move something on my screen so it's not blocking my view also. So emergence, there are several versions of emergence that have been mentioned in areas of science. And emergence is not simply in biology. Emergence exists in almost every area of, not just science, but every area of human knowledge. And these are some of the various, in philosophy, for example, this is one of the areas. So emergence is when quantitative changes in a system result in qualitative changes in behavior. This is one definition. Another definition is an ability is emergent if it is not present in smaller models, but is present in larger models. And this is a more recent definition. Another one, this is from a team that developed AI models. They were referring to emergence of intelligence in AI, artificial intelligence. So their definition, two definitions that they came up with, were that emergence refers to the situation in which an object of study exhibits properties that its individual elements do not possess by themselves. And emergence is behaviors or abilities that only emerge with scaling. And this is a very important concept, scaling. That emergence in all systems, and emergence is an element of systems behavior, system dynamics. And scaling is very important in this, in that whenever we have any kind of complex structure, it can be cells, it can be atoms or molecules, it can be people in dynamical sociological systems even. Any kind of system that has multiple elements to it, individual elements, when you scale that system to such a size that the individual elements become very large, on the order of, and again, there's no one defined size when we're talking about scaling. It doesn't have to necessarily be millions. Sometimes it's billions. So for example, with the brain, what we see is that complex types of consciousness seem to emerge when the elements, the number of neurons, and it's not simply neurons, but it's interconnections between neurons, when they surpass the small number of billions, then you start to see the initial elements of consciousness, not the types of consciousness we talk about when we're thinking of complex human consciousness, but in the simplest sense of animal consciousness, where they are aware and perceive the environment around them, and they're conscious of their being in the world, not necessarily of their own existence, but just that they are in a world, and the world around them is a physical entity. That level of consciousness emerges at hundreds of millions and usually the low billions of neuronal connections. So that's the idea around scaling, is that when a system scales, when it enlarges to such a size, a lot of more complicated, more sophisticated behaviors emerge out of that scaling. So another definition, and this I feel is really a very powerful definition, because it gets at the core and it can be extrapolated into many areas of science. And this is actually Jakob Dehaan. He is a professor of business, and he uses philosophical constructs to describe emergence. And his definition is emergence is ubiquitous. Not only can the phenomenon be observed in various systems, the term itself can be found throughout the scientific literature, from physics to sociology and everything in between. And this is very true. If you try to understand emergence, it appears pretty much everywhere and anywhere and everywhere where we look for it. Not just look, but where we see complex systems. And when we try to analyze these complex systems, it is very difficult to find a specific reason for why some of the complex phenomena emerge. It's almost inexplicable. So the common denominator of Dehaan's definition is that some property or phenomenon is observed that somehow transcends the level of the objects that nevertheless produce it. And this is a very powerful idea when we extend it into many areas. But we're going to limit ourselves mainly to biology and areas of neuroscience tonight. So emergence becomes possible when a system grows beyond a threshold, and which means when a system is sufficiently large, it exhibits behaviors that cannot be explained by the sum of its parts. The system's internals, meaning subsystems, do not explain it. So for example, in a brain, in any kind of an animal brain, individual neurons or groups of neurons cannot explain a lot of the behaviors that animal has. And it doesn't, I'm not speaking only of consciousness, but many of the more complex behaviors, such as forethought and planning. Many animals have planning. And planning cannot be explained simply by the behavior of individual groups of neurons. It's a very complicated set of advanced processes that develop, and they develop only after a certain complexity of the brain. So very small organisms, very small brains don't show planning ability. They show instinct. They rely on instinct, and sometimes that can be inferred or misunderstood to be planning, but it's really not planning. Planning is exists and appears at larger neuronal size, larger brain sizes. And emergence, so it's unexplained, it's unexplainable today. And I take a view on emergence is that it's not this magical thing whereby we can't explain it at all. And many of the things that used to be thought of as emergent phenomenon are starting to be explainable today. And I believe that that's true of almost everything that's emergent. Emergence, another way to look at emergence is it's akin to the way we think of randomness. And actually randomness in physics, for me, it's easier to explain using physics. In physics, there are actually two concepts of randomness. One is at the core of physics, which quantum mechanics is centered around, which is that there are certain parts of the universe, of the world, that are truly random and they cannot be predicted. They cannot be anticipated, and there's no information that we can have to help us to understand why something that is truly random, this type of randomness, why it occurs or how it occurs. That's the core definition of randomness in physics. The more common definition of randomness that people use, I think, is a misnomer because it really doesn't refer to true randomness. What it refers to, is such a high level of complexity in the system, meaning there's so many variables working together at interplaying together. And it is so difficult for us to understand the complexity of all of those variables together. And it is so difficult for us to know the state of that system and the value of each of those variables that because of the complexity of the information and our inability to know the full state of that system, it appears to be random. But if we had the right tools, if we had more sophisticated tools to delve into that system and to do precise measurements, to do measurements over time and to understand every part of that system in real time, that system would be explainable. And the models that we have today for many areas of the world, for example, with atmospheric sciences, with our ability to predict weather, it's actually gotten better over time. And it's remarkable given the complexity of weather forecasting. But today we can forecast weather to within a few days, within a few hours, it's very accurate. Within a few days, it's reasonable. And beyond that, a week or further than a week, it's not that accurate. And the reason is because our ability to collect information to the precision necessary where we can predict weather a week out, the precision isn't there. And the amount of data that we can collect is not there. But if we want to predict weather a few hours out or even an hour out, we have very powerful methods available today. And the models that we have are very sophisticated. And the computing systems that we have, these massive parallel computing systems, are able to do all of the processing we need. And again, the models that we have are sophisticated enough that they give us a reasonably accurate picture of what weather will be like hours or a few days later. And that's an example where in the past we used to think of weather as completely random. We couldn't predict it. It's out of our realm. But now we understand it's really not out of our realm. And by understanding that, we can understand that even predicting weather a month out or 10 years out, theoretically, is possible given the scenario of how, the quote unquote randomness of weather occurs. We can theoretically predict weather 10 years out. The problem is that getting the amount of information and having our model of weather of a planetary environment, of that model be sophisticated enough, those are the challenges. We don't have the means today to have a model that's that sophisticated, that's precise. We don't have the data and the data collection and the vast amounts of information. We would have to collect data from all across the planet for large periods of time, for months at a time, and put that into these computerized systems. And we can't do that today. That's the reason why we can't forecast long term. But if you understand the principles behind it, those principles apply to long-term forecasting. And therefore, that's one of the reasons why we understand that what we used to be called random weather is no longer considered random. And there are just many other areas of the sciences and physics especially, where because we have gained the ability to collect data and to analyze and build models, those areas that, in common parlance that are referred to as random really aren't random. In the same way, I think emergence is another one of those words where it's used in so many different ways, and consciousness, again, that's another word that's used in so many different ways that it becomes muddled. So here I'm going to focus on emergence not as this thing that can't be explained. It can't be explained today, again, because we don't have the detailed understanding of what happens in many systems, especially in the human, in animal brains. But the examples that I'm going to talk about tonight should help us to understand how emergence can be understood. And if we have enough data and the models are sophisticated enough to help us understand these systems, then emergence no longer is magical. It doesn't appear to be this out of reach phenomenon that we can't explain. So it's unexplainable today, but that doesn't mean that emergence is always unexplainable. Let me pause here for a second, for a few minutes, and just ask if anybody has any questions, or if you want to focus in on any specifics that we've talked about so far.
Hi, Sanjay.
Hi.
You done with the presentation?
Not done. Well, I'll pause right now, and then we'll have a Q&A just to make sure everyone's on board.
Wonderful. All right, I just got done, so this is perfect timing. All right, folks, go ahead and type an exclamation mark to ask questions, or you can raise your hand in Zoom. Folks, any questions? Go ahead and type an exclamation mark, or we can go to the next section, and then we will do another place for Q&A. Wait a minute, Sarah has a question.
Sarah, yeah.
Sarah, go ahead.
Yeah, I do have a question. Can you just go over what you meant with the weather predictions? I'm curious to, I want to hash that part out. My brother's a meteorologist, and he loved that his actual main job is in forecasting. So I want to see, can you just hash that part out again a little bit?
Sure,
Thanks.
Yeah, so if we look at weather forecasting, again, this is something that today is not considered an unsolvable problem. 100 years ago, we had the Farmer's Almanac, and it was basically a book that almost a sort of divination that people would kind of guess at what would happen, and they guessed months later, and because in the farming community, whether it was vital to crop survival and farmers and the community's food source. So this was a very important part of our society, our civilization, and so we worked on it for a century. And in the mid 20th century, scientists started to develop tools that have gave us a lot more capabilities to understand what weather is and how weather works. And what we call climate models started to develop at that time. And part of the reason was that because we always had things like a barometric. We could measure barometric pressure. We can measure wind speed, wind direction, temperature, humidity. All these simple metrics we could capture, and we can capture them across many geographic areas. But we couldn't go high into the various areas of the atmosphere at high altitude. We couldn't collect data simultaneously. We couldn't collect data long term. And the data that we collected was using pen and paper. People wrote it down, and oftentimes there were mistakes made in the data collection or in the data analysis. And a lot of this, there were limits on how much we could analyze the data, because all the data analysis in those days, again, this is the early 20th century or prior to the 20th century, all of the analysis and trying to understand the environment was done using people's brains. And we didn't have very sophisticated tools. Computers didn't exist then. And in the mid 20th century, we started to get computers. They began to appear. And after the initial military uses, one of the subsequent uses of the late 60s was in applying the most powerful computers to trying to help us understand weather. And not just that, we also had developments where we could use, by the time we had very powerful airplanes. And we had airplanes which could be retrofitted with a lot of measurement devices which would collect information. These planes could actually literally fly into a tornado and take measurements inside of a tornado. And then they could take measurements on the periphery of the tornado, and then take measurements completely outside. And scientists could then try to compare the various types of data using this. And these capabilities emerged over time. They became more sophisticated over time. And what happened was that this concept of modeling, which was very critical, which is very critical to science, many areas of science, but it was essential to weather forecasting, was that we needed a model, a mathematical model, you can say, but it's more than just mathematics. We needed a model to explain what are the things that cause weather. Well, first, what is weather, right? We needed to understand what weather is. So we think of weather as atmospheric conditions. And usually when we talk about weather, we're talking specifically about weather at sea level, at ground level, where people are, because we're interested in what weather is for us and for crops, or for, when we're, outside, what will it be like? But at that time, we started understanding weather is much more complicated than simply at ground level. It's at higher altitude. So we started collecting a lot of the data throughout the atmosphere of the planet. And after that, the models that they developed started to help us understand what are the forces acting on the climate and how slight effects in one area. And again, now we understand that weather has aspects of chaotic systems, which is a very complicated area. I'm not going to go into that, but that was a very important understanding that we gained. And so once we started to understand these aspects of planetary environment and started to build this model, sophisticated model, and then we started to encode these models into the computers, into computer programs. And then the next step was we would collect information, real-time information about, let's say that there's a hurricane heading toward east coast of the US. Well, scientists would go, as soon as they noticed there's a hurricane starting to form, they would send out these airplanes, or in various methods, they would collect information about the hurricane formation. And they would put that information into these computer models, these computer programs. And the computer programs would spit out answers. They basically spit out predictions. And predictions, it's not just it spit out one prediction, it spit out, let's say, 10 different versions of predictions. One prediction said this hurricane is probably going to go in this trajectory. Another prediction said a slightly different trajectory. And what scientists did was they used these predictions, and then they spent and looked at the actual trajectory of the hurricane, what it actually did. And then they compared the 10 predictions that came out of this computer program and said, well, out of these 10, these 2 are really good, and these other 3 were kind of good. And then they focused in on those 5, and then they improved it. So using what happens in evolution, they used evolutionary principles to evolve these computer programs and these models to become better and better. And over several decades, the programs now have become so reliable that now if we collect data on a forecasting on an event in the atmosphere that will happen, that's supposed to happen within a few days, the outcome that comes from these models is fairly advanced and fairly reliable. So the concept of emergence. So this isn't directly about emergence. What I was talking about was randomness, in that in the past we always thought of weather prediction as being something that we can't really do. It's kind of out there. It's too complicated for us to do. And now we've learned by following these practices that weather prediction is actually not that impossible. We've gotten fairly good at it. And I'm not talking about the weather prediction that you see on the TV news. Okay, the weather prediction that's on the TV news is actually a watered down version of it. Okay, the ones that meteorologists use, the data that they have and the models that they have are much more precise. But you have to remember that on your local news forecast, when you get a weather, when you hear the weather, what they're doing is they're looking at the weather at a specific location, usually a local airport, because that's where a lot of meteorological instruments are. It's accessible easily, or at a weather station, and they're collecting all the data. So the weather prediction is for that location. It's not for where your house is or where you are. Okay, and the generalization is that the weather forecast for that airport area is close enough to where you are that it kind of applies to let's say a 10 or 20 or 50 mile radius. Now that's the reason why a lot of times when the weather forecast says, well, we'll have rain at 2 p.m. at your house, it actually ends up being 5 p.m. because the local differences within that 50-mile radius causes it to shift in your locale, but where the airport is, the rain actually starts close to 2 p.m. It's actually fairly accurate. So these are elements of a randomness that I described, but the idea is also parallel emergence. That was the idea.
Wonderful. Thank you. Anybody else has any questions? Go ahead and type an exclamation mark. Otherwise, let's go ahead to the next section. Do you want to address, there's a comment by Katie. Would you like to address that?
I don't quite understand how you can predict randomness. I thought of randomness as surprise, meaning you didn't predict it. Yeah. Okay. So that's why I at the beginning, when I just mentioned randomness, I said there are really two types of randomness. And the problem is that in the way we talk about it, we confuse the two. And that's part of the problem with what you're raising, what Katie, you're raising, is that the core area of physics which deals with randomness, and that's primarily quantum mechanics, which at its core deals with randomness. It's all about randomness. That type of randomness cannot be predicted. Okay? That type of randomness literally cannot be predicted, and that's what randomness is. The problem is that in our verbal discussions about a lot of aspects of the world, we use the same word, randomness, to refer to things that are no longer unpredictable. So weather forecasting was one example that I gave, because it's not unpredictable anymore. It is predictable now. And we do a fairly decent job of predicting it. But the word still is used in society. And again, that application is less and less because more people are understanding now that weather forecasting is not totally random, but there's still a lot of people who refer to it as, oh, we don't really know what's going to happen in the weather. That's actually not true. So I think that's more an aspect of that our language hasn't caught up to our abilities. But yeah, what you said, that at core, randomness means that we can't predict it. Yes, and that's the quantum mechanics side of physics, which utterly is correct in that there is true randomness in the universe. But a lot of our day-to-day lives, things that we used to think of randomness, many of those things are starting to become understood better. So we shouldn't call them random, those areas randomness.
All right, Sanjay, shall we go to the next section?
Yeah. Okay. So the next part is about multicellularity, and what I want to do is I'm going to, again, I wasn't able to develop the slides too much, but I'm going to try to talk through what happened in this, and primarily, let me share screen, and then so there's really there's research that was done. Actually, this professor, a scientist, he began the research several years ago. It's a long-term set of research around multicellularity. And let me before we go too much into this, let me describe what he did. So I'm going to stop sharing just for a minute to describe, give a little bit of context of what he did and why it's important. And then I'll go into exactly what he found. So what he did is he took a very simple organism, yeast cells. And yeast cells by nature are unicellular. They really don't aggregate much. I mean, they can, but by their nature, they are individual cells, and they don't associate with any other yeast cells or any other cells for that matter. They function on their own. A single yeast cell does its thing. It lives. It metabolizes, and it gives off CO2 as many types of organisms do. And that's one of the reasons why we use yeast in baking and food products. But yeast cells are very simple to study because they have a high replication rate. They replicate in hours, several hours. So you can go through many generations. In a few days or a few weeks, you could go through many generations of yeast cells. And so that was the biological entity that this professor, William Radcliffe, he's in Georgia Tech, and he's a biologist, evolutionary biologist, and he used these cells to do studies. And he actually began looking at them. Actually, he didn't begin with yeast cells, but he began looking at the concept, the problem that he focused on when he was at the later stages of the PhD. So he's been looking at this for, I think, almost 2 decades. And what he started to do was, a few years ago, he was given his own lab and his own staff, and he started looking at trying to evolve yeast cells. Sarah asked the scientist's name. His name is William Ratcliffe, Ratcliff. And in one of the slides, I think in one of the footnotes is his info, a link to his site that you can reference if you want. So what he did is he and his team started to look at yeast cells, and what he wanted to understand is how in evolution, in the evolution of life on the planet Earth, how did organisms like bacteria and yeast cells, how did they go from being unicellular to multicellular? Okay, and because all intelligent life, all life, and most life in general is multicellular. Not in terms of the quantity of life, but in terms of the number of species of life. It seems to be at least many more species, especially if we count the number that have become extinct. There are many more that have become extinct also. So if you count all of the possible types of lives that have existed on planet Earth, multicellularity is highly prized. It's very important. And what he learned is that it actually, at multiple times throughout evolution, the process of evolution on Earth, evolution of life, multicellularity spontaneously, or we think spontaneously, automatically emerged. And he wanted to understand, why is that? Why did it happen? And how easy was it for multicellularity to occur? And multicellularity basically means a species that is unicellular, like an amoeba or a yeast cell, as it evolves over time and time and again, eventually it starts to become multicellular, meaning the cells, when they divide, they don't separate, they stay clumped together. So in human tissue, for example, when our cells divide, but they stay touching. And when one cell divides, it becomes 2 cells, and those 2 cells remain touching. And when each of those 2 cells divide, it becomes 4 cells, and all 4 cells remain touching. And 4 becomes 8, 8 becomes 16. It's a exponential rate of increase, and eventually you get millions of cells, and that starts to resemble what we call tissue. So all complex life today are built off of tissue, and these tissue eventually became organ systems, and our brain is one of those organ systems. So multicellularity is this idea that to have complex function in life, where, for example, respiration and muscles and brains, central nervous system, these things require multicellularity. That's the one mechanism that was essential for the complex types of life that we have, and especially for intelligence. So this professor wanted to understand, how is it that an organism that's doing perfectly fine being unicellular, how is it that it can become multicellular, or what is it in nature that might make it multicellular? And so he did a lot of research on this. And I won't go too much into exactly there were many areas, but I'll describe this briefly. What he did is in biolabs, it's very common to have a dish, and it can be a flask. It doesn't have to be a dish, but it can be a flask where you have an organism, a single type of organism, and you create a nice environment where this organism can thrive and it can flourish and reproduce. And that's what he did with yeast cells. He created environments where they would flourish and thrive and replicate thousands. But what he did was, every time they replicated, he took a small part of them out of the main part, and he chose the parts of the yeast cells that seemed to cling to one another. Okay, and in the beginning they just haphazardly clung to each other, and after they grew a little bit, they would fall apart and they would again become unicellular. So they really weren't multicellular, and he kept doing this for many generations of the reproductive cycle. I think initially he did it for about 5,000 generations, which took a few months. And what he found at the end of that was that the new types of cells, the new types of yeast cells that had evolved, because he was mediating, he was kind of actively changing the path that the evolution took of these yeast cells. He was intentionally changing their path. And what he found was that the new cells that emerged, that resulted after these several months, tended to clump a little more. Okay, but and so he continued this process even further and further. And what he's found that there was an ultimate limit to how much these cells could agglutinate and stay as a solid mass. And there were actually areas of physics. He brought in other scientists from physics and other areas to try to explain why these cellular masses wouldn't grow beyond a certain point. They basically would grow to just about, not quite visible, but under a millimeter in size, which if you know the size of a yeast cell, that's several million cells. It's not a small number. They would grow to fairly large, complex colonies, which were on the order of millions of cells. But even then, they would basically fall apart after a certain point of size, and he couldn't explain it. And so what he did is he modified his experiment slightly, and he started to fine-tune them and to change their evolutionary path in different ways. And I won't go into all the different things, because when he brought in other scientists, he started looking at the metabolic cycle of how oxygen plays a role in the cellular membrane. Many areas that he looked at, because for two cells to clump together and stay clumped together, the cellular membranes have to have certain characteristics. So he modified his experiment to try to basically select for him. He was literally selecting for certain characteristics. And as he did that, the newer cells that evolved were getting better and better at clumping, and they would actually grow it to larger sizes. But after a certain point, and this is one of the key things that happened, was after a certain point, they kind of started growing into huge structures, huge cellular at the cellular level, meaning they started to become visible to the naked eye, larger than a millimeter. And some of them grew to almost a centimeter in size, which is huge if you think of a colony. And you have to remember, these are still basically the same. The DNA hadn't changed that drastically. They were still yeast cells, unicellular yeast cells, but they had been evolved. His team had evolved these yeast cells to start to show characteristics of multicellular organisms. And so that's what I'm going to go into. And so let me switch to a slide now, and we'll go into that a little bit. And again, I'm going to have to talk through it because it's complicated in what he did, but hopefully people will understand exactly what. So let me actually, the first slide is a precursor to some of the things I talked about. So first, emergence of multicellularity. Around 3.9 billion years ago, roughly, 500 million years plus, or not 500 million years minus, it's more plus. It's estimated that the first types of unicellular life originated on Earth. They were not like cells that we see today. They weren't complex. They had a bilipid layer. They didn't have DNA. The initial ones didn't even have RNA. The later ones, which could replicate, had RNA. But the initial ones were somewhere across between what we think of a virus today and what we think of a precursor to a cell. I won't go too much into this, but they were very, very simple organisms. And then those simple organisms evolved more into what we think of today as unicellular organisms, like a yeast cell. And again, this happened around 3.5 billion years ago. And then after that, we started to get what's known as LUCA, the last universal common ancestor. And again, this is not the first cell that existed. This is the first cell that survived. There are many other cells that came before LUCA, but all of them had become extinct. So LUCA is the first cell that still exists today, or its DNA still exists in the cell lines today, in all of us today. So LUCA is the first cell that we can trace back to, which shows all of the signs of life that exists today on planet Earth. So it's the first cell line, you can say, that we can say we originated from. but it's not the first cell. There were many cells before that. All of those other cells went extinct. And again, this was not sorry. This shouldn't. This says 3.9 billion. This was close to 3.5 billion years ago. But again, this isn't. These estimates are very rough because it's so difficult to many different models that are used to do these estimates. And Luca basically developed into all cellular life today, especially the multicellular life that we are and animals are. And multicellularity evolved out of this sequence. And multicellularity is believed to have developed at least 10 times in our evolutionary past in these 3.9 billion years, into 3.5 billion years. And they developed in separate ways in separate areas of the biosphere of the planet. And they all happened in different ways. So plants, for example, have it in the organisms, in small organisms, for example, Plactocines, they're another type. So anyway, so these are there are many types of multicellular life that originated. So this is one of the slides that. So what I'm going to do is I'm going to describe the things that he found out as part of this experiment. And this experiment he's built onto it and done a lot more work since then. But I want to highlight some of the key things that he found as part of this. And so what happened? What he found was that, and what the slide shows is that you'll see four different colors, four different clusters, you might say, and each of these colors represent a different type of yeast cell. Okay, so in the top right corner you'll see gray, and those are the original yeast cells that he started his experiment with. And the experiment that I'm talking about is not the original experiment I mentioned before. He expanded on that experiment many times, and so this experiment it's gone on for at least 2 years. I believe more than that, but at least 2 years, where he's modified it in such a way that what's come out of this is that a model that he's formed and the evidence that he's shown really seems to give us a very good understanding of how multicellularity may have evolved. Out of the 10 times that we know of, that we estimate may have happened, this seems to match the model around those 10 times that it happened. And it's a fairly good approximation. Again, we don't know if this is exactly what happened, but in the experiment, because he's collected good data and analysis is good, we actually can see this happening. And anybody can replicate his experiments and they'll see similar processes happening. And because of the way he did the experiment, there are so many things that we see that help us to understand, the different stages that the cells went through. So one of the ideas that came out of this is that in this particular experiment, and again, the experiment that he was using yeast cells, and if a different scientist starts with a different type of unicellular organism, probably aspects of what they find will be slightly different from what Professor Radcliffe found. For example, he found that the ancestor yeast cells evolved into 3 distinct types of new cells. So a different scientist who does the same experiment with a different type of precursor organism, they may find they evolve into 4 or 5 or only 2 new cells. They might not, or they might, also evolve into 3 cells. But the fact that it's 3 is not important here. The point that's important is that the evolution allowed the cell to not just evolve into a new cell, it allowed it and made it evolve into 3 distinct types of cells. And each of these cells are important. They all evoke very important elements or characteristics that when they work together, when these 3 types of cells, yeast cells work together, they work together to make the organism start to become multicellular. And I'll describe a little bit about what these 3 types of cells are. So the original, and in the multicellular organism, after the 2 years of evolution, after he allowed it to evolve for 2 years, changing, every replicant, every replication cycle, which lasts a few hours. So his team was doing this constantly, daily, every few hours. It was a lot of work. And after, close to 2 years, they started seeing these 3 distinct cell types that emerged. And one of the cell types, and actually they started seeing these even before the 2 years had elapsed. They started seeing a lot of different unique features. So one of the first things that they saw before they saw the differentiation into three, they saw that the green region here, the growing type, what they first noticed were most of the yeast cells were getting very, very large. They were really growing into very large cells. And in a sense, if you think about that, that's one way that a unicellular organism can become multicellular. If you have a lot of large cells, instead of a cell being a specific size, let's say a micron in size, if a cell becomes 10 micron in size, meaning it balloons literally like a balloon to 10 times its size, well, the organ, and you have 5 of those cells, right? 5 of those cells that are each 10 microns becomes 50 microns. The radius is going to be less, but it becomes a fairly large organism. Again, it's only comprised of 5 cells, but if you compare it to its predecessor, which was only 1 micron size, right? The comparison will be that this multicellular organism is huge. So that was a initial finding that he had, was that these cells adopted this mechanism of ballooning, literally ballooning, becoming super large, to allow, because that gives more surface area if they're larger. So the surface area also helped them to cohese with each other. And so that was a early technique that the DNA evolved in these cells to allow them to clump together. Now, the problem is that these large cells were unstable. And soon after, he started seeing that they would grow to a certain size, and then they would kind of fall apart into small, tiny chunks. And then those chunks would grow even more, and they would fall apart again into smaller chunks. And this kept happening over and over. They would grow and then almost like shatter. They're not literally shattering, but they would declump. They would fall apart into small clusters, and they would grow again. And this cycle repeated over and over. So then he modified the experiment a little more, and he started to see, eventually he saw at the end of 2 years that there were 3 distinct cell types that came out of this. One, the green ones that we talked about that are growing, the ones near the center, the top center, which are the blue cells. And these cells had a very unique type of cell wall. And again, this is not surprising now, because cell walls are important for cells to cohese, to agglutinate and to stay touching to each other. So the cell wall membrane evolved in certain ways. I won't go into detail with how, but they evolved in the right way that allowed them to clump together. And the third way was what's known as programmed cell death. Apoptosis is the official word. And what happened was that out of the colonies, the large colonies that developed, a certain percentage of them, and again, in here we can see the percentage roughly, but the percentage varied. A certain percentage of them basically automatically died. They basically automatically self-destructed. That's programmed cell death. Their DNA had elements built into it, and that exists even in multicellular life today, even us, in our brains even. Programmed cell death exists in many, many organisms and many of our organs. So this is not something unique that you saw in cells. This is something that exists today in most biological tissue. And it just so happens that this also evolved in these yeast cells. But the purpose of this evolution, he couldn't explain, he couldn't understand until later, the further findings. So these are 3 distinct areas that his team saw initially. And they were trying to explain it, they couldn't explain it. The second thing that they saw was that, and in this diagram, the top diagram, it represents the early version of his experiment where these yeast cells would agglutinate. And in this case, we see 3 in the center that are larger. Those were the ones that were ballooning. And they also had other cells that were not ballooning. But this is the type of clustering that they were doing. And this is the type of clustering that they would basically fall apart. And these are known as the snowflake yeast ancestor cells. And the reason why you call them snowflake yeast cells was that because when you looked under a microscope, their shapes sometimes resembled snowflakes. They had hexagonal structure, or like a snowflake has 6 spokes or 6 intricate patterns. They seem to show these types of patterns. So he chose this term snowflake yeast cells. But the problem with at this level of evolution was that they were limited in how much they can grow. They fell apart after a certain size, no matter what he tried, until he again talked to other scientists, and he modified the experiment, and he created an environment, which again, what he did is he talked to scientists who understood and have studied the early primordial environment of Earth, because at that time it was believed that the Earth didn't have oxygen, but the nuances of what he learned, he applied into his experiment, in that it's not that early Earth didn't have oxygen. It didn't have oxygen, then it had oxygen for a little while, and then oxygen again was depleted, and then it came back. And when he changed his experiment in that way, to mimic it that way, these yeast cells evolved further with further abilities, and eventually to the 3 types of cells that we talked about. And the bottom of this slide is important because one of the things that happened is that these cells, they developed the ability or the feature to elongate themselves. And this was critical to what he ultimately found. They were no longer spherical or spheroid type shapes. They were basically long shapes. And the way that they clustered, the way that they agglutinated was also unique. And this kind of shows a structure of this. And you might say that this looks like a root structure. It does resemble root structure in some sense. A lot of organisms today, especially plant roots, and even, believe it or not, dendritic structure in our brains, the structure of how neurons are. Not that these cells are doing what neurons do, but the shape of axons in the brain resemble these. Axons are much more complicated than these, but you start to see similarity in what these yeast cells are doing, and much more complicated, sophisticated, and future evolved systems in biology. You start to see parallels in these at a very early age. I mean, these yeast cells had not really evolved much. He had evolved them a few months at that point. So in a few months of evolution, we start to see emergent properties that exist even today in very complex tissue and animals. So this was important in one of the findings that he came up with, and he continued. And so these are diagrams of the types of these structures that exist in many types of life today, in trees, in sponges. These are in the center is some types of tissues. What's deemed as D, I'm not sure what, but these are fibrous tissues. Again, there are many types of tissues in the organic world that exist. E, I'm not positive, but E is, it seems to be, these are not cellular, but these are the remnants of in a lot of tissues. What happens is cells grow, and then when the cells die, they leave behind a structure, a physical structure, similar to what happens in corals. And that I believe that's what it is. But basically, this is showing the structure that the last slide showed. The bottom of the last slide. It's giving examples of these that exist today throughout nature, throughout the biological diaspora. They exist today. And these are all a result of multicellular activity. The next, this is an animation that, and this is another element that he found, which was phenomenal. When I read about this, it really blew my mind. And it's difficult to understand or even to see, but I'll have to explain this. And this, I left this on a loop. Hopefully the animation is playing so you can see it. And what he did is he basically took 300 seconds, which is 5 minutes of elapsed time, in the center are the yeast cells. And there are many, these are not countable. They're probably tens of thousands, and the size roughly is about 2 millimeters across. So this is a significant size when you think about yeast cells. Actually, it wouldn't be several tens of thousands. It would be, I believe, millions of cells. I don't remember the size of yeast cells. I can't extrapolate that right now off the cuff, but it's a significant number of cells that we see here. And there are 3 things that I want to bring your attention to. One, so let me explain what we're seeing. The center part, the orange part, are yeast cells, many yeast cells that have agglutinated together, and they're remaining together. They're functioning, in a sense, as a multicellular organism. Also, what we see is on the periphery, you see these orange dots, and you see that they're moving kind of inward. And if you look carefully, they are moving inward, but you also will see that there are other more diffuse patterns of dots that are moving outward. So you see both influx, and you also see an exflux. You see both. If you look carefully, hopefully the video is clear enough that everybody sees that, but it's important to see that, and I'll explain what that means. And then the third thing that is important to see is, and especially it's in this area where the mouse is right now, what you'll see is kind of like, and literally what it is it's movement of fluid. It's fluidic movement. So if you think about what circulation is in complex life, right? Our blood flow. What our blood is doing is it's a compartmentalized channel in a capillary or a vein or artery, a larger structure. It's a self-enclosed cavity through which liquid, a specific type of liquid plasma with a lot of other types of molecules and organic compounds, they flow in this channel, and that's what our blood vessels are. And that is very complicated in complex life, but something very similar to that has started to appear in this type of organism. Okay, and it's evident in the area that I pointed to, kind of at the upper end of the center where the mouse is. You see this channel, and again, this organism is three-dimensional. The video is two-dimensional, so you have to kind of extrapolate. But you can see that the area along this channel is kind of sparse, and it kind of tends to shift because the organism is moving in space, and as the fluid is passing through it, the way the camera captures this image gets affected by the amount of fluid. So there's just some complexity in how we can do imaging on this. But it really is a fluid channel in there. And the fluid channels are not just that area, but there are many different areas of this organism. So basically what happened, what he found, was that at a certain point, this organism evolved the ability to move liquid into the center areas of this organism. And if you think about it, this is vital to the survival of multicellular organisms, because in a multicellular organism, if that didn't happen, then the cells that are in the inside are kind of landlocked. They're locked in, they're surrounded by other cells. And if you think about it as a three-dimensional organism, almost like, if you think, the simplest way is a sphere. If you think of it as a solid ball of cells, the cells all the way at the center have no access to the outside, right? And they're surrounded by other cells. And they're surrounded in terms of getting respiratory, because cells have to metabolize. Cells survive by metabolism, so they need some type of gas, either oxygen or CO2. They need some type of gas to permeate through their membrane into the cell structure. Excuse me, and then they metabolize, and then there are waste products that come out of that cell and they're excreted out. So cells that would be the center of an organism would lose that ability, or their ability to do that would be severely diminished because they're surrounded by other cells. And the partial pressure, getting a little bit into the biology and chemistry of it, of these cells would prevent them from surviving. So the center mass of these cells would literally die, and this cellular system would either fall apart, where it would die, or it would kind of open up into a flat structure. It would not be, and there would be a limit. We would see a limit to how large they could grow. And so that is what would happen in all cells, unless there was a way that fluid and air could get inside of this organism into the various areas within it. And that's what this video captured. That's what this part of the experiment captured, is that these cells actually evolved this ability spontaneously. He was not selecting for it. He was not doing anything. He didn't even expect this to happen. His team didn't expect this. They were quite literally surprised by this also. But he did a phenomenal job. They did a phenomenal job of capturing these types of elements to show that this is, in fact, this does take place in these groups of cells that they're growing. And this has been replicated many, many times. It's not just a one-time occurrence. This happens in pretty much all of the future generations of the evolved versions of these yeast cells, where they built, if you think of a capillary network of blood vessels in a living being, right? This is a very early precursor to that. They're not blood. It's not blood at all. It's simply moving fluid in and out. And the fluid purpose is to allow cooling, heat exchange, energy exchange. Also, it allows the elements of metabolism to both come in and leave. So earlier when I described these tiny dots all around the periphery, they're moving toward the center. That's because these cells, and again, this is sped up. This is very, very sped up. But these cells are pulling in CO2, they're pulling in oxygen, and they're metabolizing, and then they're releasing waste products. So earlier when I described these elements that are moving toward the center, also if you look closely, there are clouds. These are more diffusive. They're not, clumps as much, but because a lot of the waste products are not clusters. They're like a cloudy liquid, you can say. But if you look closely at it, if you look, for example, at this area, you might see there are waves, waves of these structures. I mean, even in this area, they're going completely outward, and this is a characteristic of the biochemistry and biology that's happening, whereby the difference in pressure within the cells and within this organism and the difference in pressure outside of it are causing this movement inward and outward of different molecules. And this is phenomenal, because what this is showing is that these cells, which don't have a purpose for these capillary actions, actually evolved this ability. So hopefully people understand, and it's surprising to you also, because this was remarkable what he saw. This is another element that he saw, is that these cells, and this is a time lapse of the growth, and this shows the elongated structure. And here it's not as elongated. This may have been one of the earlier versions, but if you see, compared to a sphere or a circle, these are, some of these are like the initial ones. This one, if you look at the beginning, when it comes, it's very, very long. Compared to if you think about its width versus its length, it's 3 to 4 times longer than it is wide. That's a significant change in its growth pattern, in its DNA. Basically, its DNA is causing it to do that. And the magenta versus the blue is just a way of differentiating between the first two cells that began. The first two cells, one was labeled magenta and the second cell was labeled the bluish cyan color. And you see the proliferation of these, and the fact that each of them are proliferating at a similar rate. So it's not that one was proliferating more than the other. Many things that they had to look at in this, but this image gives us a lot of insight into what actually is happening in these yeast cells. There's another video. This was actually done, I believe this was done later. This was done in March of this year. And these are using fluorescent biomarkers. So this is not using normal types of imaging, but this is a longer time span, and where we can actually see inside the cells many elements that are also happening, and structures that are developing inside the cells, how they replicate, and it's much more sophisticated understanding that this is giving us. But basically, what this shows us. So let me go back to one of the earlier. I want to go back to this slide, because this slide really is very important. This and the slide that shows the water channels, the fluidic channels inside the organism. Those are the two, I believe, two of the really, really powerful ideas in this work. And in this slide, again, 3 types of cells that evolved, and each of these cells are very, very distinct from the original base or ancestral yeast cell. They're significantly different from the ancestral yeast cell. And I suspect that if he continues this evolutionary process, that these cells will differentiate even further beyond just these 3 types. And if you understand what that means is that these 3 cells types together are allowing what we call tissues to develop, because these clustering, at the beginning, they're clumps. But if they grow even further, they will start to form effectively what we call tissue in biology, in biological studies and biological sciences. Tissues are cells which not only are multicellular, but there are specific specializations of cells. And in the brain, we've talked about this a lot. Brain doesn't have just neurons; it has many, many other types of cells, also support systems. Astrocytes, many types of cells which help neurons to survive. And this is actually what we're seeing here, is that the base cell, which is the green cell, these are the core cells, but also these blue cells are just as important, because these blue cells you can think of as glue type cells. There are cells where the cell walls are different enough and they kind of cling to the green cells to keep them all together. And the red cells are, I'm not sure what color, anyway, the reddish cells are important, are believed to be important for providing these the channels, the fluidic channels that we talked about, because for these fluidic channels to open up, one way would be if the cells literally moved. But the problem is that these cells don't have cilia. They don't have a mechanism for moving in space. They can't move themselves. So one of the mechanisms would be if certain cells literally die, leaving a vacuum within the larger structure. And that's currently believed to be what may be happening in these, where a large number of cells along certain paths die. That's one thing that allows these channels to form. Other things that this program cell death allows is that it allows this tissue to literally replicate into larger tissue, because if you have fracture lines along a tissue, that tissue can proliferate in much more complex ways. If you don't have any fracture lines, if the tissue basically continues along the same path without any differentiation, you would basically have the blue and green cells all to themselves and nothing more. And effectively, the blue and green cells are very similar to each other. So you wouldn't have much differences in these cells. But the fact that these red cells are there and they create these vacuums or these holes throughout the tissue, then those holes, through evolution, through further processes of evolution, those holes can have a fourth type of cell or a fifth type of cell or additional types of cells. If this experiment continues, I believe probably we will see fourth and fifth and further types of differentiation in these cells, which give rise to more sophisticated behavior in these tissues, which we see in normal life. So that's one of the powerful things in all of this that is seen. Let me stop here and just pause to see if there's anything that needs, if anyone has any questions. And we'll go into the reason why this is important for emergence after this idea is I hope everyone understands what this is saying. And if you don't, feel free to ask it. No question is too simple. I think this is a very important element of what we've talked about, what we talk about in this area. So I want to make sure people really understand this.
Thank you. Thank you, Sanjay. Folks, go ahead and type ! In the chat or raise your hand in Zoom in order to ask questions or make any comments. Folks, don't be shy. No questions.
Hopefully that's a good sign that makes sense to everyone. So let me ask a question then.
Yes.
Just general, anybody, if they want to answer, you can raise your hand, ! You can raise your hand. So given that these cells, in his experiment, he found that these cells are differentiating. And these are yeast cells. So yeast cells by themselves don't really have function other than just to live and survive. Does anybody want to take a guess that what might be another type of cell that arises in these? And again, there's no right or wrong answer here, and we don't know. There's no answer literally because his experiment is continuing. But I just want to provoke the idea in everyone here to see, what you think about it, and maybe what you project might happen. What type of cells do you think? What type of features in these cells do you think might come about? DLJ, I think, Reese.
DLJ, go ahead.
Yeah, are you hinting at the eukaryotic revolution here, from prokaryote to eukaryote?
I mean, that's a much further stage, yeah. Eventually we'll get, I'm not going to go into it today, but that's one example of what would happen, what could happen. Yeah, but I'm leaving it open-ended completely, where anything, what do you think an organism like this, if you think of the organism that we saw, that orange blob, which is kind of building this network of fluidic paths within itself, what's the next thing it could do? What benefit would it have? What might it need? I mean, for example, one of the things is that it would need to evolve into a larger structure. Let me put that as a prompt, that growing larger is what he's selecting for. Okay, that's the one characteristic that he's selecting for. By tuning his experiment, he's choosing the ones that grow larger. So that's a given, that they will grow larger. So for a larger organism, what would help it be viable? What type of new cells would help it be viable as it grows larger? And there's no one answer. There's many things that can happen. But I just want people to explore. I think Madeline, she raised her hand.
Madeline, go ahead.
At a guess, it would either be something for motility, or it would be a void for digestion, like a little gut, proto-gut.
Yeah, excellent. Both are very good answers. Yeah. So just to give an example, so motility, ability to move, and these right now, this cellular blob has no way of moving. It stays there, and it's kind of at the whims of the water, of the fluidic currents in there. It kind of flows with it, and it has no way of moving around. It has no way of changing its orientation to turning or anything like that. And it has literally no information gathering from the external world. It has no neurons. It has no ability to sense or react to the world around it. It simply lives. It simply kind of stays there as the metabolites allow it to function and grow. That's it. It just grows. So if it evolves the ability to move itself, that will help it to develop into more sophisticated animals, which can actually move to find their food. So that's what Madeline said. I agree completely, is that not to say that that's the next thing it would evolve, but eventually it would evolve the ability to move itself using things like cilia. Cilia are a very basic structure that we see in many types of even unicellular organisms. They evolve a cilia fairly early on because it helps them to navigate and move around. And this type of organism would definitely do that eventually, because it would give powerful abilities to it to move and find food, or to stay in a place, or to evade a predator, things like that. And the second thing that Madeline mentioned is forming a gut. So another way to think of that is that right now these organisms are not able to really do much with metabolism. They are relying on gaseous exchange and any nutrients that are in this fluid nutritional matrix that they're living in. And it's up to the scientists to kind of provide the nutrients for them. And it just absorbs through osmosis. It absorbs into their, through their permeable membrane, cellular membrane. And then the waste products go out, and it comes in and goes out, and comes in and goes out, and that's their life. Just over and over, and then they replicate this process, nothing more. But if they had to live in a more complex world where there are other organisms that they could feed off of, then you need a pathway where that food could come in. So you need some type of a mouth structure, you need some type of a stomach structure, esophagus, where you can channel the food. And it might not be esophagus; it might just be that you have a central cavity. You have an opening, which acts like a simple rudimentary mouth, where it opens, it engulfs a organism, digests it in a way, and the mouth opens and it releases from the same cavity. It doesn't have to have two cavities. So so those types of things would be things that emerge. So what we start to see is that something this simple, it's showing us a pathway for complex life, how everything we see today on planet Earth has evolved. And it gives us a direct line of sight, a direct connect the dots from simple unicellular life to the most complex life that we have today. That's why I believe this work is so powerful, because it gives us a very easy to understand and very easy to replicate. I mean, he did this in a few months. Any other scientist can do the same thing in a few months. And I suspect there will be, in the next few years, there will be many other teams that work on and modify the experiments in many different novel ways. His team is doing it in many other areas also. I'm not going to go into those. But we can learn so much about these sophisticated processes that came out in all of life, starting from the most primordial part of evolution, going from a single cell to complex, really complex life, going from primordial singular cells to single structure of a matrix of simple cells, and then going to a tissue that's a little more complicated, and then going to what we think of as organs, right? Or not even before organs, into a more like a hydra. If you think about a hydra is a multicellular organism that has these tentacles, and it has a hollow cavity. It doesn't even have a stomach. Basically, its tentacles aren't actually moving food. What they're doing is they're moving the liquid and they're channeling liquid through its central cavity. And any organisms that happen to be in the environment kind of flow through the central cavity. And if they come in contact with its membranes in that central cavity, there's exchange of energy, there's exchange of nutrients. It literally digests some of those organisms. And so Hydra is it's still a very simple organism, but we can see very clearly how a Hydra would evolve from this. And from a Hydra, we can see how more complex organisms would evolve, the precursor ocean-dwelling creatures, etc. So it's a really powerful element. Now, the part where emergence comes into this, that it's very important, is that, and for me, the fact that the multiple types of cells evolved, not just multiple types of cells randomly, but these multiple types of cells evolved with very discrete functions that work together. It's not that one type of cell evolved and it kind of does its own thing, and another type of cell evolved and it does. These three types of cells in that chart, in that table, in that chart, sorry, those three types of cells work together. So they evolve to work together. That's very important to understand. So there's some type of dynamic that happened in the evolutionary process, whereby the DNA of one category of yeast cells, again, they started from the same cells, one category of yeast cells evolved in one direction, another cell line evolved in a different direction, but both their evolutions were somehow linked. And the third cell line also evolved in a third direction, but all of their evolutions were linked in symbiotic ways so that they all work together to help this tripartite organism, this three-celled organism, to evolve sophisticated lifelike, or not lifelike, it was life even as a unicellular, but evolved into very sophisticated complex life. You know, the emergence of complex life literally emerged out of nothing. The experiment did not do anything that would have caused that to happen. All that he was choosing for, all that he was selecting for in these cells was largeness. That's all he was selecting for. He didn't look at them under the microscope to see characteristics. He simply looked at the physical size that they grew at, a rough physical size. He chose the ones that were larger and separated them and let them grow. And the ones that were smaller, he kind of didn't let them. So every generation, he just picked the large ones, let them grow. Pick the large ones, let them grow. He didn't focus on what it was about them or what was happening. And automatically he saw this. So that one aspect, that these three elements of these three types of cells evolved, it's a very surprising aspect that this happened. And secondly, at least for me, the fact that these fluidic channels spontaneously arose, and in what actually, when I saw this, what it brought to mind for me is that, and this is in the news a lot, it's been in the news a lot, that's the reason why this visually came to mind, is that when you look at a human fetus or animal fetuses, in the first few days and weeks, they also have this very similar structure evolve. And problem is that a lot of non-scientists believe it has to do with blood flow and heartbeat. It's not about that. It's not a heartbeat. It's not blood flow. It's similar to what this is, what this study has found, that it's basically the internal cooling of this massive cells in a fetus, an animal's fetus, and the ability of that fetus to grow larger because it allows nutrients to come in. If it didn't do this, that fetus literally would not be able to grow. It would halt. And so that's what came to mind for me, because I saw that parallel directly. And if you think about it, that is every organism today in its embryonic state, when it starts as an ovum, gets fertilized, and then zygote replicates into a multicellular organism. It's exact same path. So his experiment simulates the path of every zygote, every living being, every living zygote, multicellular organism that grows today. It replicates that exact same process, but he did it, in a unique experimental way. So it's phenomenal.
Thank you, Sanjay. Next up is DLJ. DLJ, go ahead.
Oh yeah. So I was going to give you an answer to the previous question, but I think you've moved on from there. So
No, please, go ahead.
I think I'll put it as a question. So I was going to pick up on when you said moving away from danger, because presumably the cell, because it's still single cell, right?
Well, no, it's not a single cell, it's multicellular.
Oh, it's multicellular now.
It's
Okay.
multicellular, yes.
Okay. So, so presumably some parts of that multicellular system, which is where we get towards cell differentiation, right? Some of them must detect that risk or that danger, right? So I'm guessing that before light sensitivity, there was probably pH sensitivity. So moving away from an acidic environment, that kind of thing. So what's the idea about how some, not necessarily all of the cells, but some like differentiation, right? Some are becoming detector cells. How does that work?
Right. So we can go in any number of directions here, okay? Because this experiment sets a stage for explaining pretty much everything that we see in life. So you mentioned detectability of light, of the environment, detectability of pH. That's also a sense of detection of the environment. Let me give a different version of pH. When we talk about the Hydra. Now, Hydra, I don't believe have this, but a little more advanced versions of feeding organisms, what they do, and this we see in our own biology, is that our stomach has literally hydrochloric acid, which is a very it's very acidic. It's a very caustic element which our body produces. And not only that our body produces this acid, but the cells in our stomach and the linings around it and the mucosa are grow. Our DNA does this. It grows and remains that way to give us a stomach that can have a highly acidic environment. So a precursor to mammals, a precursor to even reptiles and fish who have some bit of acidity inside their digestive tract, a very early precursor would have developed into the internal parts of that organism, would have developed cells, again, cellular differentiation, would have developed some cells in the central part, which when they give off the waste products of its metabolism, they would have given off either higher pH or lower pH. There's no right or wrong here. It could have gone where the digestive system was more basic in terms of chemistry base, or acidic. It could have gone either way. Okay, there's no right or wrong. And you can model an experiment where one organism would evolve into having chemical bases as being the basis of its digestion, another organism using acid. We tend to use acid to digest our food because we get it in that direction, but just as easily could have gone the other direction. Now, there's a little more complex. I won't go into the complexity. There's a reason why it didn't go to the base using bases, because it's a little more, it creates a little more challenges with the organism. But anyway, these initial organisms could have gone either way, that when they're releasing their waste products, they're releasing more acidic products, at least some of the cells to the center of it, where you think of a gut, where you think of a stomach type of thing, right? Those initial cells might have released more acidic waste products. And those acidic waste products, the other cells around it would have not pushed it out. So another level of complexity would have been that this organism not only has developed this gut, but then this organism also has other cells which cause some things to move into the gut and other things to move out of the gut. And so a third or fourth level of sophistication of specificity of the cells would be some cells would allow food-like particles to come into the cell, and other cells would allow waste products to move out of the cell, but not to allow the acid to move out of the cell, to move out of the central part of this organism, right? So if you let this organism evolve far enough out, you start to see that the proliferation, differentiation of its cells becomes so unique that within the digestive tract, you have some cells that retain food particles, other cells that retain the acidic digestive compounds, and other cells that push out the waste products. So in us, our waste products are in sweat, in our blood, in urine, and our digestive system, and also our respiration. Okay, all of those are parts of our waste. But our waste system has become extremely complicated. Our cells give off waste in many different areas, and we push it out of our body using many, many different ways, hundreds of ways you can think of. But this organism, I mean, the one we saw doesn't do any of it. I mean, it does very, very rudimentary one or two of those things, but you can extrapolate where it'll do more complex things after just a few thousand more generations. It may evolve the ability to have a functioning stomach, and to have a functioning stomach, it would have to have the ability to retain food particles. It would have to have the ability to retain this acidic juice. It would have to have the ability to push out waste product while not pushing out. And this gets into, it's not intelligence, but it gets into biological sensitivity, biochemical sensitivity, that it's pushing out waste products which are not acidic. So again, and I think somebody in the chat mentioned hydrophobia. So actually, hydrophobia is something that exists from the first cell onward, because all cells, the cellular membrane, is composed of a bilipid layer, which has one end which is hydrophobic, the other end is hydrophilic. So that hydrophobia comes with the territory from the beginning of cellular life. That automatically is there. So DLJ, I don't know if I mentioned, you talked about pH and I forget something else you talked about. It's light sensitivity, right? So light sensitivity is another aspect but light sensitivity would be much more complicated, because you need a lot of different types of cellular structures, a lot of different types of cells that have to evolve. For example, you have to have cells that are close to neurons, because the light has to send the signal to something like a precursor brain. So that, I think, would happen, probably hundreds of thousands, if not millions of generations into the future of an organism, because sensitivity to light would require, it would require the ability to process information. Photons are information. So it would have to understand light here, no light there. And that dichotomy of being able to distinguish between light above and no light below, just to differentiate between which is upward and which is downward in the ocean, that ability would require a very simplistic neuron or structures of neurons. But yeah, that would eventually happen.
The word understand there is a very loaded term, because that implies comprehension as opposed to competence.
Right. So at that level, the level we're talking about, yes, there would be no understanding at all. It would simply be reacting. It would be the order of what a light switch, literally what a light switch in our house does. When a light switch, we flip it, it reacts and lets electricity pass through. That's exactly what these initial organisms would have done. When light hits the sensory receptors that it has, that photon would get converted to electrical impulse. It would shoot through the axon to a cluster of cells, and those cells would, direct the energy toward cilia, causing it to move in one direction, to move toward the light, let's say. So it'd be a very simple thing. As soon as light shines, it moves toward the light. As soon as light goes away, it stops moving. Something that simple.
Folks, you're welcome to comment on anything in the presentation you found interesting. If you just want to talk about what you found interesting, you're welcome to do that. If you want to go ahead and ask questions, you can do that. DLJ, go ahead.
I was just going to flippantly comment on how did vampires evolve then? Because they move away from light, not towards it.
I think that's a rhetorical question. We'll leave it at that.
Thank you, DLJ. Anybody else? Any questions? Vampires or otherwise?
Or zombies? Because I believe zombies are supposed to be not alive, undead.
So in that case, let us take this opportunity to look forward.
Yeah.
So Sanjay, what are you planning to do for the next for the remaining part of the year? What kind of topics are you thinking of?
So I mean, the list that I gave in the intro to this talk, that's been in the back of my mind for a long time. And I think this is one of the first times where I've actually listed it out. But each of those areas are very complicated, and it's even this topic, I don't think we've got into fully. I may need to cover other aspects of it, because in terms of understanding complex things, what I've realized is that we each need to develop an intuition around what it means, our own personal intuition of what it means and how we think about it. So here I gave examples just to help everyone to gain this their personal intuition around it. That when you see this, and I'm trying to use visual methods because that really helps us to focus. I mean, I talk through, but I suspect that the words that I was using in the beginning to explain the experiment probably was more difficult for people to conceptualize what was happening, and the video or the images helped to concretize the ideas a little more. So I think that that's one of the things that I want to do, because when we each have an innate understanding, a personal understanding of what it means for an organism to evolve and evolve sophistication, and for that sophistication to come automatically. I mean, it seems to be magical right now, but later concepts will be where we go into, no, it's not magical. You know, when I talked about weather forecasting, there was a reason why I gave that example, or, you know, there are other examples I could have given, but weather forecasting is something that most people now believe is not magical. It's in the realm of no longer, magicians, meteorologists, or not magicians, they're scientists. They use pure science in a didactic way. They use it to help us in our daily lives. And by the same token, the concept of emergence, the last talk that we had, there were a lot of discussion around, is emergence a real thing, or is it even useful to talk about emergence? Because the word emergence has so many different versions of it. And a lot of that talk was around trying to narrow down and navigate through these different meanings of it. That's why in this one I tried to narrow down to the concept that emergence, at least the way I see it, you know, other scientists do, but the way I see emergence is not that it's referencing this mystery. It's not referencing mystery. It appears to be mysterious right now because we don't have the data. We don't have the view, the micro view. So for example, that cluster of cells, we could see that there's fluid flowing through it. But just to give an example, if he continues these experiments, or other teams, if they continue the same experiments, except they also use more sophisticated measuring techniques. And they use these measuring techniques not just at the time scale that he used time scale of seconds, 300 seconds, right? But if these future teams use the time scale of milliseconds and microseconds and even nanoseconds, right? If they start to look at the evolution of a single, within this multicellular organism, right? Each of the cells are changing every few microsecond. Okay, that's the time span that we have observable changes. So if they're able to take, let's say, 100 cells in this multicellular organism, 100 cells, okay, and if they can measure many aspects of each of these 100 cells at a time scale of every millisecond, okay, that's a lot of data. And they continue doing this for, let's say, 10 seconds. That might be gigabytes worth of data. That's a lot of data to collect at the precision that we need. Well, if they do that, and enough teams do that, we will start to see how these channels emerged, right? Because right now it's mysterious. It seems magical that these channels kind of just popped out of nowhere. But if we have measurements of exactly how, and all of the different cells, and what we might learn, I'm speculating here, but what we see are 3 different types of cells. What we might learn is not just 3 cells. There might be 5 or 6 different types of cells, but 2 or 3 of those cells emerge temporarily, and then the mechanism of apoptosis, cell death, caused those 3 other types of cells to disappear. So we never read, because this scientist was using a large time scale. He was using seconds. So we don't have a resolution fast enough to see those cells disappearing. He simply saw the vacuum, right? He saw that these cells are gone, but he didn't see what happened in the small time scale, the millisecond time scale. So if we have further studies at more precise techniques using precise tools, we will see more sophisticated processes which will help us to form a model of this area, which right now appears mystical, magical, right? But we will start to see and understand it's not mystical, that in the beginning we might see 5 types of cells that proliferate. 3 of these cells are critical for the multicellularity. Another 2 are critical for forming these channels. And another one might be the one that provokes the apoptosis, the death of some of these cells. So we will start to learn that. And then from that knowledge of these 6 types of cells, other teams will do further studies, which delve further in, just like what we did with building the models of atmosphere and predicting weather. The same thing, that what we'll be able to do in that is to understand that the emergence that we think of it as emergence today. And again, I prescribe the notion that emergence simply means it's too complicated for us to understand. It's not that we can't understand it. I personally believe anything that's emergent, we eventually can understand, but we don't have the tools today. We don't have the sophisticated tools, but in the future we will develop those tools. So emergence is a word that's important for us because it helps us to understand and label these things which are too complicated, what we used to call random, right? And in a sense, this phenomenon of these behaviors in this multicellular organism, these yeast organisms, seems to be random. That why is it that this cell does this, and why is it that in this location it built this cavity and not another location? It seems to be random to us. We can't explain why it happened in this area, not this area. But as we delve further, we realize it's not randomness. It might be that there is a size of the cells, that certain cells of a certain size who have a certain number of neighbors, that those are the ones that turn into this cell. And other characteristics of cells turn into another one of these cells. And there may be certain patterns that we start to see that causes that. So what we think of as random today, then we realize it's not random. It's actually driven by underlying forces. So that's a lot of what science is, deep science. So what I hope is that people start to see these things, and we build an intuition that in the brain, a lot of the things that we think of as unexplainable, they're really not unexplainable. From the mid-1900s, we've learned so much about animal brains that many things that we earlier had false notions of or we misunderstood, now we understand fairly well. We can still go much further, but we have a lot of understanding that, and so I personally believe that consciousness is one of those things that also is emergent. And the reason why the last talk we looked at AI is because used from that vantage point, it appears that consciousness or precursors or appearances of consciousness, again, they're not conscious right now. These AI systems are nowhere near, and I don't think they will be for many years. Some people say they never will be, but that's to see. But we're starting to see the pathway, the emergence of behavior that's conscious-like, the more complicated areas of consciousness, especially of noticing and reacting. For example, I'm not talking about the consciousness of knowing that we are conscious and knowing that we are a being that can think about oneself, think about our own awareness. That's very, very high level consciousness. These AI systems aren't there yet, but they are aware, or I shouldn't use the word aware. They can give the semblance of communicating the idea that they know about themselves. If you give it a prompt, most of these LLM models, if you give it a prompt, if you type in a question saying, "What are you?" Okay. First, it figures out we're talking about it. It figures that out. That we're talking about it. It is a subject, right? That it itself is a subject, and that's an important idea to have, the idea of consciousness. Now, I don't believe these systems have consciousness in that way. They don't understand themselves as an entity. What these systems today are doing is they're using their understanding of language, and the way they manipulate language to us appears because they're trained on human language, which all human language has behind it the veneer of consciousness, right? Any person that communicates with a notion and characteristics of consciousness. That's why our language has that embedded within it. And these precursor systems today, these AI systems today, give us similar mimicked language, which we interpret as having a lot of these human-like characteristics. But
okay, I have to tell you something on a lighter side here,
yeah.
And then I want to make a comment about everything that you're saying. On this point about language and ChatGPT. So I gave ChatGPT 4 a prompt to say, write a poem about your future in the style of a Shakespeare sonnet. And it went on, "Oh, ChatGPT, thou art a wondrous thing, a marvel of technology and mind. Thy neural network, swift and true, doth bring answers to questions of all humankind." 4 paragraphs, beautifully done. Then I said, write a scathing. So I took the entire text fed it in again and said, write a scathing critique of this poem in the style of a jaded and cantankerous New York critic. And it went ahead, what a trite and unoriginal piece of writing is this? The author seems to have no imagination and instead relies on tired, cliched phrases to express their admiration for ChatGPT. And then for the third, I asked it to say, again, fed in the critique and said, rebut this critical argument systematically and coolly, calmly, in form of a poem written in Shakespeare's sonnet. So it went ahead and said, thou sayest this poem is trite and bland, unoriginal and lacking in wit, but I say that criticism is unplanned and based on assumptions that do not fit. And it goes on, each of these is like multiple paragraphs, but there is just on the light side to kind of just refit it, but it's really interesting. I'm the most interesting thing about ChatGPT is that I've started suspecting when people talk to me is, okay, wait a minute, are you just being a ChatGPT or are you actually a human being? It's kind of going back and evaluating what human beings are doing. Coming back to what you're saying, I think what you're doing here is really, really fascinating, because if this is a frontier of science that you're dealing with, and the way you're dealing with it is actually in the great scientific tradition, because at different points of time, different fields were in this state where our knowledge about it was not particularly good, and chemistry was in a state, biology was in a state, physics was in a state at that time, and we had all kinds of semi-concepts or something to point to an effect we didn't really understand. We had to work through that terminology. We had to design experiments. We had to be patient. And we had to be, firstly, true to the observations that we were seeing, even when we couldn't explain them, and saying that, look, this is what I see. I don't know why this is, but this is what I see, and kind of, remain true to that. Then we had to kind of carefully bring up our instrumentation, whether it was measurement of time by Galileo or things like that, to have some way of figuring out, okay, if you're going to measure something like gravity, the pendulums are not going to cut it. You need to find something else. And so you kind of have to kind of up the acuity of measurement on one hand, and then you have to use kind of multiple of these things together. Then on the top of that, you have this fascinating thing about experiments, where you say, okay, now let's actually try to build, see how, have these different organisms evolve and see what happens. Then have some kind of a very simple strategy like size, for example. I think that's just brilliant of finding that. And there also, it's like, what do you choose? This is like the heart of it. Like I remember reading Optics by Newton. And at the end of the Optics, there is this short section called Queries. And his questions, I was just floored by his questions, because if you ask the right question, you cut the Gordian knot. It seems like a completely messy thing, but these people, it's like a good question just kind of separates it out. So, okay, if I separate light out, I see that light is separated out. I've seen prisms. If it is separated out, what happens if I combine it back? And what does that show about the nature of light? So it is an adventure. It is science itself is an adventure. And and this area right now is one of the areas which is at the it's experiencing this amazing growth. And the beautiful thing about it is that so many people are working on it so well. So it's like this mass of scientists and all kind of communicating with each other, going back and forth. So it's just a wondrous adventure, and you are doing a very good job showing the the grandeur of this enterprise. So thank you very much. I'm really grateful.
No, thank you. I mean, one thing that I want to make sure that these are beneficial to people and it actually helps. So I wanted to ask everyone, I mean, do you think that this is the right approach to helping explain and understand neuroscience in these areas? Do you think there's something else that we should do? What just general ideas? I mean, because these are all very difficult ideas to portray. One of the most difficult aspects for me in putting any of the presentations together is, I mean, the idea I can verbalize, but that doesn't mean it's going to, the way I think of communicating or informing is there's an idea in my mind. I want to get that same idea or a very similar idea to someone else's mind. And all I have are words or pictures or things like that. And a lot of times it's very difficult to do, especially if there's no basis that we share in understanding. So for example, when I was talking about the types of cell differentiation, I think when I gave the example and Madeline, I think she helped a lot of this when she described an organism that has a gut, that has a stomach, because we all know most organisms have, the ones we know about have a stomach, that I realized that I can use that to show that the stomach we understand, but we might not understand that the stomach has to have multiple types of cells within it. Some cells that produce the acids, some cells that produce a mucous membrane to protect itself from the acid. Because in our stomach, that acid is very, very acidic. Hydrochloric acid literally erodes metals, so it easily will erode our cells. So, is there a better way, or do you think there are other things that we can do in these talks?
Yeah, folks, this is, please give us feedback. Let's start with Madeline, but we'd love to hear from many people. What is it that you're getting? What else would you like? What are the difficulties you're having? What can be done to overcome them? Please go ahead and type an exclamation mark or raise your hand in Zoom. Madeline.
Yes, thanks, Shrikant. This is fantastic, Sanjay. I would not change a thing. The only thing if you could, I know you really can't due to time limitations, but if you could put the references up like a day or two before, we could look at them. Also, what about my placozoans? I love my trichoplaques. What happened? Are you going to cover that guy next time?
Well, we might have to do a third. But one thing is that what I also want to do is not to make it boring for it, because everybody is not interested in the same area. So I might do the third one a little bit later, a few months later. The difficulty is that the information we've covered, we may lose track of. So there's I've tried both versions and there's no right way to do it. If I do a third one next month, it will help the people who've been following on. But people who either didn't follow on or aren't interested in this, it wouldn't help them. So I don't know. I mean,
thank you. Anybody else wants to comment on how this series is going? What you would like to see? Any thoughts?
Oh,
Folks,
also,
go ahead.
sorry, I just wanted to say also, I find showing the videos while you're speaking is very helpful.
Okay. Wait, you said slowing the video or showing the video?
Showing them, just showing them is very helpful.
Okay. You mean the speaker's video?
Yeah, the one from the paper from the Radcliffe lab that you were showing, things like that are very helpful.
Okay, so any kind of animation or videos, yeah, I agree. I think that helps to concretize a lot of the ideas, but it's very difficult to find things that are not under some type of IP protection. It's very difficult to find a lot of those. So, but I try to.
Thank you. I think Jyoti has a question.
Yeah. So chimera, she's asking about chimera. So chimera, there's actually many different, that word also has many meanings. So there's a concept of chimera in chemistry. I am assuming, I'm not sure exactly which part of Kimura you're referring to, but I suspect you're talking about Kimura as an animals, organisms.
Jyoti, would you like to elaborate on that question so Sanjay knows clearly what you're asking?
Yeah, hi Sanjay, how are you? Great presentations.
Hi.
I'm listening to them. I'm under the weather. The allergies took the best of me. Chimera or Kimera, whatever you want to call it. It's when people like parents may have an offspring who is totally different than any one of them, the characteristics. And usually they say that this child has probably got cells from different sources. I never understood that. If you have any inkling about that.
Well, I mean, I'm not an expert in this. This is basically embryology, but I can give you a little bit of information that I've read about. And you're specifically referring to biological chimera. So chimera, the broader concept of chimera is any multicellular organism, any form of life, and we can think of a person or an animal, any that has characteristics of more than one being. Okay, and that can mean two different things. Okay, it can mean that it has characteristics of two different beings, meaning it can has characteristics of a human being, as well as, let's say, a lion. Okay, or any two organisms. So a chimera, the traditional definition of a chimera in mythology, but in biology it still retains, but biology expanded on it, is that you have like a centaur. People are familiar with a centaur, half horse, half human. That's a chimera. But the more modern definition, and this is the part about embryology, is that when a fetus is growing in the womb, it's a complex environment. Many things can happen. Typically, nothing unusual happens, but every once in a while, a small percentage of the time, unusual things happen. And there are many types of things that can happen. So, for example, one of the things that can happen is that you can have the zygote, the cell, the initial zygoblast that divides and divides. But at a certain point, early in its division, it splits into two. And this is similar to what happens in a twin, in an identical twin. But what may happen is that these two blastocysts, they split, but then they remain kind of bound slightly. And as they grow, and this may be because of, again, many reasons why it can happen. But for example, one thing that can happen is that one of those blastocysts, they both continue growing as separate organisms, as separate beings. So just as what happens with twins inside the womb, they're two babies and they grow independently. You end up with two, hopefully functional, completely separate babies. But if things don't go well, and many things can go wrong, you can have many combinations of this. One of the combinations could be that one of those groups of cells, while it's separated, it stays embedded within the other group of cells. And so what happens is, up to a certain point, one of the babies is growing, and the second baby continues to grow inside of it. So literally one baby is growing inside of the other baby. But after a certain point, because of the dynamics and they're competing and there's all kinds of things, the inside baby stops growing, and the outer baby continues growing, and it becomes a baby and it becomes alive, and the mother gives birth. And it may be that the doctors find that inside baby, or they might not. There have been cases in science where after that baby became an adult and they went for some kind of medical procedure, that's when the medical community realized that there are these cells which seem to be different and abnormal, but it's a small group. It's a small, and they're there inside this person's body. And you can't take it out because it's too destructive. They cannot end up leaving it. So that's an example of a chimera. There are many other examples. The differentiation and the separation may be in organs, whereby a chimera may be that they have organs which either don't develop or they have multiple organs. For example, they might have two different livers, or their liver splits in two parts, or one part is liver-like and the other part is kind of merges into the kidney and it disrupts. And a lot of times when this happens, the baby actually dies because it's too disruptive. The body doesn't work properly and they die. But there are enough slight few cases where the baby is able to survive, but later in life it develops some disease. at which point it's found. So these are examples. It's basically the problems have to do with embryology, where the embryo is developing in utero and there's something that goes wrong during the development process. But it goes wrong, and if it goes wrong far enough, the baby dies. But if it doesn't go far enough in the wrong direction, then the cells that remain literally remain in the baby that continues growing and that is born. So hopefully this explains what you're asking.
All right.
Thank you, Sanjay.
All right. Thank you very much, everybody. Really appreciate all of you folks. And Sanjay, this is wonderful as always. So we'll see you in a month. And we've got all kinds of things coming up. So Thursday, tomorrow, is going to be Prophet by Khalil Gibran. Friday, Jang, Avanique, and Joe are doing a series called Designing Your Life. There is an author who is speaking. It's a new, Jang keeps on bringing different people from different fields to talk about designing life. On Saturday, we are doing Asian philosophy. On Sunday, we are doing Buckminster Fuller. On Mondays, we are doing collaborative poetry. On Tuesdays, we are doing Art of War. And next Wednesday, it's going to be Phil on different theories of history. And he's going to be talking about structuralism in history. So we have quite a few things going on. So please join us. Good night, everybody. See you. Thanks, Sanjay, again. Bye.
Thank you so much.
Good night.
Bye.
Good night.